much. I am a professor of pharmacy practice and infectious disease at Creighton University. I'm the pharmacist that rounds with the consultation service here at Bergen Mercy Hospital in Omaha. And I gave this presentation last Friday to the ID Fellows. And so Dr. Horn wanted me to give this to all of you. And so we'll go through and talk about treatment of gram-negative bacilli. So here's my objectives that I'm going to review. So we'll recently talk about some drugs that are useful for resistant gram-negative pathogens. We'll talk about some of the advantages and some of the disadvantages associated with those agents, and then where we can apply the clinical trial data to the treatment for multi-drug resistant organism. And so we'll talk about ceftazidime-avibactam, ceftolozane-tazobactam, Cefiderocol, Sulbactam-durlobactam, Eravacycline, and then there are some others that are in the pipeline, and so we'll talk a little bit about those. So it's pretty common knowledge that the CDC has issued both urgent threats as well as serious threats to the use of multi-drug resistant organisms. And so under the classification of urgent threats, carbapenem-resistant Enterobacteriaceae or Enterobacterales is an urgent threat. And then under the category of serious resistance is multi-drug resistant Pseudomonas aeruginosa, besides some of the other ones. So we're going to concentrate primarily on CREs, or carbapenem-resistant Enterobacterales, and then also multi-drug resistant Pseudomonas. And so here's what we're talking about. Drugs or organisms that are resistant to more than two antimicrobial classifications or classes basically are considered multi-drug resistant isolates. And so we don't keep track of multi-drug resistant isolates, but just based on our own antibiogram that's published here within CHI, we have approximately 2,500 E. coli's that would be considered multi-drug resistant. We probably have a little bit over 200 Klebsiella pneumoniae that are multi-drug resistant, and we probably have the majority is our Pseudomonas aeruginosa that are multi-drug resistant in our facilities. This antibiogram that is developed by the micro department actually compiles all of the isolates from all of the surrounding Omaha area CHI hospitals. And so this isn't just exclusively to Bergen Mercy, it's also associated with other hospitals around the Omaha area. So if you look at the Ambler classification for multi-drug resistance, there's 4 different classes, A, B, C, and D. A, C, and D are associated with a serine beta-lactamase. They can either be penicillinases for class A, or they can be carbapenemases. And so the most common carbapenemase in the class A category is the Klebsiella pneumoniae carbapenemase. And then the penicillinases include some of the older TEMs and SHIVs. CTXM would probably be the most common ESBL isolate that we have, certainly in our area of the country. And we have some relatively good inhibitors of the class A beta-lactamases. The class B are basically metallo-beta-lactamases. They require a zinc ion, and they're basically carbapenemases almost entirely. So this is probably the most common New Delhi metallo-beta-lactamases, as well as the IMPs or the imipenem carbapenemases and the VIMs carbapenemases. These can be inhibited by aztreonam, in addition to maybe adding that to maybe some other agent that we'll talk about. The class C carbapenemases are basically cephalosporinases. They're resistant to clavulanic acid, and these are, for the most part, AMP-C associated resistance mechanisms. And we have some resistant beta-lactamase inhibitors that can help for those. And then the class D beta-lactamases are primarily oxacillinases or carbapenemases, and these are the OXA-associated carbapenemases, and we have a few beta-lactamases that will help with that. So the carbapenem-resistant Enterobacterales, the KPC was initially discovered in 2001 from a patient that was hospitalized in North Carolina. The KPC is probably in the US the most predominant multi-drug resistant isolate here in the US. The OXA48 like carbapenemases are concerning from patients that stay mostly in Europe, like France and Spain and things like that. And then the class B beta-lactamases are the New Delhi metallo-beta-lactamases, and they are concerning from patients that come from India or Southeast Asia. This is probably the most notorious carbapenemase, and fortunately we have seen only sporadic cases here in the US, but we're obviously concerned about this because it's only a plane ride away from having these organisms become endemic to different areas within the country. So let's talk a little bit about what we're speaking about. So we have a 61-year-old female that's admitted to our hospital. She has a ventral hernia repair, so she gets exploratory laparotomy in September of 2022. She recovers from that repair and goes to a long-term care facility. And then in the care facility, she develops fever. And a urine culture is drawn, and it shows this Pseudomonas aeruginosa isolate with the susceptibility pattern. So sensitive to amikacin, resistant to cefepime, intermediate to both ciprofloxacin and gentamicin, resistant to levofloxacin, very resistant to meropenem, resistant to piperacillin-tazobactam, and then sensitive to tobramycin. So probably this is the most common type of isolate that we see. And so this is the one that obviously concerns everybody. So we know that ceftazidime is resistant to hydrolysis by many of the older beta-lactamases, including the TEM1s that's common in like H. flu and Moraxella catarrhalis. But it's not stable against ESBLs or organisms that produce the AMP-C enzymes. And ceftazidime is not not sensitive to organisms that produce KPCs or metallobeta-lactamases. So avibactam was added to ceftazidime to protect it against class A and class C serine beta-lactamases. So that would be the ESBLs and the AMP-Cs. Ceftolozane-tazobactam is 8 to 16-fold lower MICs compared to ceftazidime, imipenem, and ciprofloxacin. And the effectiveness of ceftolozane-tazobactam is limited by the production of ESBLs, beta-lactamases. So stability to hydrolysis by AMP-C can be dependent on the level of expression. And so if you have a high level of expression, you aren't going to be able to inhibit that isolate. So tazobactam elevates the activity against the beta-lactamase producing isolates, as well as ESBL producers and some inhibition of AMP-C derepressed Enterobacter and Citrobacter strains. It's still susceptible to hydrolysis by metallobeta-lactamases as well as KPCs. So we have that whole across the whole class that we're going to talk about. Meropenem is resistant to hydrolysis by many beta-lactamases except the KPCs. Vaborbactam is a novel boronate beta-lactamase inhibitor that's been added to meropenem and protects meropenem from destruction, but it may not work very well against the class D carbapenemases. And then if your carbapenem, so vaborbactam doesn't increase your susceptibilities associated with both your metallobeta-lactamases as well as your oxacillinases. Imipenem-relobactam is reported to be useful for KPC resistant to ceftazidime avibactam as well as meropenem vaborbactam. However, in the study that this was produced in, it was only 15 isolates, so maybe we need some more isolates to actually make that comparison. So avibactam restores ceftazidime activity against various gram-negative bacilli and Pseudomonas, so it expands the spectrum of activity for ceftazidime for multi-drug resistant isolates. Patients treated with ceftazidime avibactam for Pseudomonas bacteremia or pneumonia were more likely to develop resistance. And so that's something that we have to watch for. The study by Van Duren was the first prospective study that looked at clinical outcomes in carbapenem-resistant Enterobacterales infections, either bacteremias or pneumonias. The mortality was 8% in patients that got Cas-Avi and was 33% in patients who got colistin. So the adjusted all-cause mortality was significantly lower. However, again, we have to look at this microbiological failure rates during therapy. So ceftazidime-avibactam resistance was approximately 30% of all the microbiological failures. And so this is something that we're going to need to continue to determine whether or not resistance develops during therapy for patients that get ceftazidime-avibactam. For ceftolozane-tazobactam, Hayter et al. Just looked at a number of multi-drug resistant Pseudomonas and found a 71% clinical efficacy. However, in those patients, 14% of them had developed resistance during therapy. And then we know about the multi-center randomized open label Tangle 2 trial that compared the efficacy of meropenem-vapobactam to best available therapy for serious carbapenem-resistant Enterobacterales infections. Here, in this particular study, the majority of patients receive meropenem-vapobactam solely as monotherapy or with another active agent, whereas the best available therapy may have used anywhere between 2 or 3 agents for the serious carbapenem-resistant infections. And the results demonstrated a significantly better clinical cure rates for meropenem-vapobactam, as well as 28-day mortality. So several in vitro studies have shown meropenem-vapobactam would be useful in patients that have ceftazidime-avibactam resistant KPC producing isolates. So as a summary then, ceftazidime-avibactam is useful against Klebsiella pneumoniae carbapenemases. Ceftolozane-tazobactam has added value against MDR Pseudomonas, and so we primarily try to use that only for Pseudomonas, although it may have some other activity. And then meropenem-vapobactam may be useful if your isolate develops a resistance to ceftazidime-avibactam. The thing that's most important to remember is that if the isolate is resistant to the carbapenem, either meropenem or imipenem, the beta-lactamase inhibitor doesn't protect it from that resistance mechanism. So just as we showed for this particular case, we have a meropenem resistant isolate. So in that particular case, you wouldn't add or you wouldn't recommend meropenem-vapobactam because it wouldn't protect that that meropenem from that resistance mechanism. Okay, so let's talk about some newer options that are available. So Cefiderocol is a novel siderophore cephalosporin that has a catechol moiety that contributes to its potent antimicrobial activity in gram-negative isolates. The siderophore is an iron complexing protein that chelates with the ferric ion and utilizes the bacterial iron transport system to penetrate the outer membrane of these organisms. And so this particular drug then doesn't use the regular porins to get into the periplasmic space to inhibit the penicillin binding proteins. The 2 grams every 8 hours would be the standard dose for patients with normal renal function. And the probability of target attainment with a 3-hour infusion is very good. And based on animal models, You get greater than or equal to a 1 log reduction in bacteria if you have a time of the fraction of the free concentration greater than the MIC, greater than 40 to 70%. So here's some of the susceptibilities that were submitted to the FDA for Cefiderocol approval. You can see for carbapenem non-susceptible Enterobacterales, 97% efficacy or susceptibility. For carbapenem non-susceptible non-fermenters, that's about 95, 96%. Real good against Pseudomonas, as well as Acinetobacter baumannii, and then certainly very good against Stenotrophomonas multifilii. So here's some of your MIC distributions associated with some of the different mechanisms associated with the resistance. You have KPCs, you have IMPs, VIMs, NDMs, and then you have a whole list of oxacillinases. And you can see that the break point of 4, you can see greater than 90% inhibition associated with this drug. Here's a pretty complex slide looking at pharmacokinetics. Here you have on the x-axis MICs associated with the different isolates, and then you have the frequency of the MIC distribution. And so at an MIC of 4 with 2 grams every 8 hours, you're still well above 90% probability of target attainment. And so that's actually very good for inhibiting multi-drug resistant isolates. So here is the complicated urinary tract infection results for Cefiderocol versus imipenem. And you can see in the modified intention to treat analysis, you can see very good efficacy compared to imipenem. Microbiological efficacy was also very good. And then microbiological response also very good. So the credible CR study was a randomized open label study to assess the efficacy and safety of Cefiderocol against best available therapy for carbapenem resistant gram-negative isolates. Patients were randomized to Cefiderocol or best available therapy in a 2-to-1 module. 45% of the patients had VAP or HAP. 31% had bloodstream infections and or sepsis. And 24% had complicated urinary tract infections. And you can see here that the endpoints at the test of cure visit by infection site, as well as for clinical cure and microbiological cure, was pretty good for all of the different sites or types of infections, as well as for the microbiological eradication. So Cefiderocol is a new siderophore cephalosporin. It uses a Trojan horse for intracellular transport. That's why it's called Phe-Troja as the trade name. So iron Phe and then Troja for Trojan horse. It's active against multi-drug resistant isolates, including ceftolozane, tazobactam, and Cas-Avi resistant isolates. It's active against multi-drug resistant Acinetobacter, as well as Stenotrophomonas and Burkholderia cepacia. It's approved for chronic or for complicated urinary tract infections, and there's questionable use, whether we can use it at other sites of infections. And so more data is probably needed to make that comparison. So now let's switch gears a little bit and talk about Sulbactam-durlobactam. So this is primarily a drug that was fast-tracked and approved by the FDA for carbapenem-resistant Acinetobacter baumannii, or CRAB infections. And most CRAB infections have a horizontal acquisition of a class D oxacylinase type carbapenemase. They can have other resistant mechanisms in addition to this, including acquisition of class B carbapenemases. You can lose your outer membrane protein, or you can modify your ABC resistant efflux pump that may be associated with fluoroquinolone therapy. So Sulbactam was actually developed by Pfizer as a sulfone beta-lactamase inhibitor. It's co-formulated with ampicillin and commonly used against CRAB infections with other antibiotics. But primarily what's working on the Acinetobacter baumannii is the sulbactam, not the ampicillin. The mechanism for its usefulness is not well known. It could be due to its effect on inhibiting the penicillin binding protein 3, or it could be subject to degradation by beta-lactamases in Acinetobacter baumannii. So combining sulbactam with a novel broad-spectrum beta-lactamase inhibitor could be a sound approach. And so that was hypothesized. And so then pharmaceutical companies went to try and find that beta-lactamase inhibitor that they could couple with sulbactam. And so durlobactam is a broad-spectrum diazobicyclooctane non-beta-lactam beta-lactamase inhibitor. It's similar in structure to relobactam, and against carbapenem-resistant Acinetobacter baumannii, it's highly potent inhibitor of Acinetobacter cephalosporinases and class D beta-lactamases, including carbapenemases of the oxa family. So here's some results of a phase 3 multi-center randomized controlled trial where patients with CRAB infections had received sulbactam-durlobactam plus imipenem versus colistin plus imipenem. And the primary endpoint was the percent of patients that had 28-day mortality. And you can see in the sulbactam-durlobactam group, the mortality was less than 20%, whereas in the imipenem colistin, it was about 34%. So roughly a little bit, almost half associated mortality. So Eravacycline is another tetracycline that's a fluorocycline. It's synthetic. It's similar to tigecycline. So tigecycline is 4 times more potent than doxycycline, and Eravacycline is 4 times more potent than tigecycline. It's been shown to be active against most bacteria that express the TET targeting efflux mechanism and other ribosomal protection mechanisms, and it doesn't seem to be affected by beta-lactamases because that's not the mechanism for action. It's approved for complicated intra-abdominal infections, but it's not active against Pseudomonas or Burkholderia. So it's active against crab infections. It's highly bioavailable after oral administration. It has very good metabolic stability after metabolism in the liver and has a low potential for drug-drug interactions associated with concomitant drugs the patients are receiving. So in the complicated intra-abdominal infection, where Eravacycline was compared to ertapenem, this is the IGNITE-1 study. It's a randomized, double-blind, non-inferiority trial. Patients received Eravacycline, a milligram per kilogram every 12 hours, and that was compared to ertapenem, a gram every 24. And no differences in whether or not in the clinical cure rates. However, in the IGNITE 2 and 3 study, it did not show non-inferiority compared to levofloxacin for complicated urinary tract infections, and it did not show non-inferiority compared to ertapenem. So, and people theorize that the reason for that is the lack of penetration into renal parenchymal tissue, and so that's why that didn't have too good of an effect. Okay, so another cephalosporin beta-lactamase inhibitor combination that's newly approved is Cefepime-enmetazobactam. It's approved for complicated urinary tract infections. It's 2.5 grams every 8 hours, over 2 hours for 7 to 14 days in patients that have normal renal function. The beta-lactamase inhibitor is a new penicillinic acid sulfone beta-lactam ACE inhibitor targeting Enterobacterales ESBLs. And so it protects cefepime from degradation by certain serine beta-lactamases, such as the class A ESBLs. And while the structure is similar to tazobactam, a key difference is that this beta-lactamase inhibitor is a zwitterion, so that enhances its potency. It's able to get into tissues easier. It does not reliably inhibit class A carbapenemases like KPC, and it's shown to have limited inhibitory activity for class D beta-lactamase inhibitors. So Cefepime-enmetazobactam has demonstrated in vitro activity against contemporary MDR enterobacterales that have the presence of some beta-lactamases and ESBLs. So that's something that's also available currently. So in the clinical trials, there was a phase 3 randomized double-blind trial that primary endpoint compared to piperacillin and tazobactam was a microbiological modified intent to treat. And you can see the efficacy associated with overall patients in patients that had ESBL producing bacteria, as well as in patients that also had bacteremias. So on the horizon is mirpavidine. It's a protein epitope mimic that targets the outer membrane protein, targeting antibiotics. It primarily looks at LPS transport protein D in the LPS biogenesis of the outer membrane of Pseudomonas aeruginosa. It's been studied in ventilator-associated pneumonia due to Pseudomonas. Of the 12 patients that were used with confirmed Pseudomonas, 9 had multi-drug resistant, and the clinical cure was 91%. 28-day mortality was 8% compared to a 20 to 40% expected mortality from Pseudomonas. So more to come on this particular agent, and it'll probably be talked about at ID Week coming up here in the fall. Aztreonam-avibactam is active against metallo-beta-lactamase producing bacteria, but is hydrolyzed by Ambler class A and class C beta-lactamases. There's a revisit study that's currently ongoing looking at Aztreonam-avibactam with or without metronidazole compared to meropenem with or without colistin for the treatment of HAP, VAP, and complicated interabdominal infections due to gram-negative bacteria, where the treatment options are limited or there's no treatment options. So that's currently ongoing. And then we have several others that are being produced or investigated: Cefepime, meropenem, and Cefiderocol, and then there's a whole bunch of beta-lactamase inhibitors that just have numbers so far. And so I think that that is going to be interesting. So the summary that we have for multi-drug resistant isolates, here we know that ceftazidime-avibactam is good for class A carbapenemases. Ceftolozane-tazobactam may be useful for carbapenem-resistant Pseudomonas aeruginosa. Cefiderocol is pretty good against all of these different multi-drug resistant isolates. Eravacycline with KPCs and crabs. Upiravan for carbapenem-resistant Pseudomonas, and Aztreonam-avibactam may be useful for these two classes of drugs or isolates. So with that, I'll stop here and let me stop sharing. And then if there's any questions, I'll be happy to try and answer them. Don't see any questions in the chat. So I guess I can give you, if there's no questions, I can give you back some time in your day, about 20 minutes.